Recording. So tomorrow we have a quiz, correct? Yes. What's your quiz on? Glucose and fructose. So the video, all the notes from Monday, correct? Posted already. So you know I'm going to pick 6 or 7 questions. You know they're going to be applied. They're not going to be what's glucose, what's fructose, none of that. All right. So we talked about different structures. We talked about carbon having 4 bonds. We talked about how when you see, and I think we had some really good discussions in class, that when you see these lines and they stick out, that these lines are showing you where the carbons are, that each of these lines connecting is a carbon. And that there are always 4, and based on that knowledge, you can decipher, you can break down what the molecules are, or even build them the way they're supposed to. We talked about the different kinds of macro, now we called it, someone mentioned macromolecules. So there's macromolecules in biological terms turn out to be polymers. Except for lipids. Lipids are technically not polymers. They are macromolecules, they're large, but we're not going to have repeating units. A polymer is a molecule made of repeating units. And we talked about the fact that that's a polymer. Poly means many. There's 3 prefixes. There's a dimer. Di means 2. So 2 units to make something that's made of just 2 units is a dimer. There's oligo. You might see oligosaccharide, oligopeptide. Oligo means a few. And there's poly means many. You might see that in the book. You might see that. You might hear a lot of that. You certainly will see it in college, and if you do any research in a lab, you're going to see these prefixes. So macromolecules are big. They're put together with something called dehydration synthesis. Which is not going to be on the test because that's not from Monday's notes as far as I remember. But what will be on the quiz tomorrow, let me highlight this in yellow as not being on the quiz. Is that fair? Yes. So this is not on the test and this is not on the test, right? But the other stuff is, this other stuff is. I don't know if it's going to be on the test. I haven't written it yet, but it's not in the notes for Monday. So it's not, this yellow is not in the notes for Monday, so it's not going to be able to be on the test. We did talk about the long chains, and notice I'm using the word chain, of sugar molecules. And I'm going to use the word monomers. Long chains of sugar monomers. I think this is going to wrap up carbohydrates for us. We also talked about the fact that sugars are made of carbon mainly. I'm going to put mainly. You can attach anything to anything, okay? And your body does. Your body attaches sugars to proteins, sugars to lipids. But mainly sugars are made of carbon, hydrogen, and oxygen, specifically in a 1:2:1 ratio. So always have the same number of oxygens and carbons, and you have twice as many hydrogens. And you built the molecules on Friday so you can see why that has to be, because carbon has to have 4, and every carbon has an oxygen, and that's just the way it is. We also talked about on Monday the idea of some of these, let's see. So what were these long chains of sugars that we talked about? We talked about glycogen. We talked about cellulose. We talked about starch, right? What different kinds of starch were there? There were 2 kinds of starch. What were they? Potatoes are where they're found. What were the 2 different kinds of starches?
Alpha and beta.
Alpha and beta were the linkages. But there's 2 words.
Is it glucose and amylose?
It's amylose is one. Amylopectin. Now here comes a spelling problem. Amylose, amylopectin. So it's beta, beta linkage, and amylopectin. Alpha. Is she right? Is she right? Amylose is alpha 1,4. And amylopectin is alpha 1,4 and alpha 1,6. Ultimately, what does that mean? What about amylopectin? That amylopectin is branched. This is talking about how they're linked. How they're linked changes their shape. Their shape changes what enzyme will work on them. Some things will do well with amylopectin, they won't work with amylose. Some things will do well with amylose, but not amylopectin. Some things will do well with glycogen, but not those others. We have the ability to digest glycogen, which is how we store our sugar, and amylose. What about amylopectin? Is that something that you can digest easily enough?
Yeah.
Yes or no?
You can digest amylose. Why not? Wait, so 1 to 6 are the prime like carbon connections like in the in like the shape? When it says alpha 1 through 6? Yeah, 1 prime, 2 prime, 6.
The 1 and 6, the numbers, are the carbon number that they're attached to. The alpha or the beta is a conformation. So which direction, what angle, is it up or down, that kind of thing. I don't really want to get into it because it's not really that deep, but it is something that you should know that for instance, amylose is water soluble where amylopectin is not. So let's take a look at what might the differences in starch be.
And isn't like if there's alpha it's more curled than beta, which is straight and cellulose?
Cellulose, it's beta, helps it be straight, but also that makes it straighter, but also there's no branching. Right, so you get the best of both worlds when it comes to looking at. So both are digestible. It's that amylopectin. Well, which one was more easily digestible? No, I'm asking. I know this. This I know, unfortunately. Amylopectin is more easily digestible. Amylose is not as digestible, but still is. And the reasons are surface area. And why is it more surface area available to get at the bonds? Because they're what? They're branched. They're open to attack. So your enzymes can attack here and here and here and here and here and here. Versus amylose, it's spiraled, so it can only attack at certain places at a time. So this is obviously going to be easier to digest. Now, is that a good thing? If you're going to eat starch, is that a good thing, ladies? Is that a good thing? That you break it down quickly? Yes or no? And give me reasoning. So breaking down is good, so you get more sugar quicker. Ideally, if it was just that, you would say, okay, that's good. But what happens, you get a sugar rush in your blood, and your blood does not do well. Your body doesn't do well with a lot of sugar in it. So you have to pump insulin into your blood to get the sugar out of your blood. So there's a lot of things have to happen if you have a sugar rush. That's why eating candy is not the best thing. Eating simple sugars is not the best thing. Amylopectin is a harder to digest sugar, so the sugar will more slowly enter your bloodstream. So you probably are going to want to eat more amylose than amylopectin. They're both going to be starchy foods, but you probably want more amylose in your diet than amylopectin. Why?
No, you're fine. You're right.
You don't want sugar. The reason I know this well is because I'm diabetic. You don't want sugar rushing into your blood. That's not something anybody wants unless you're having a diabetic ketoacidosis or you're having a diabetic incident, like you're passed out or you're going into a coma. Other than that, you don't want sugar rushing into your blood. High blood sugar is bad. Can we just say that? High blood sugar, bad. Amylopectin is easier to digest, so when you eat a starch product that has a lot of amylopectin and not as much amylose, you're going to pump your blood full of sugar. That's not good. Where amylose takes a little longer to get into your... Not because it's harder energetically, just literally the structure. What's the key that I want you to get away from out of this? And I think that they were talking about yesterday too, by the way, is to look at the structure. Structural differences influence the function. Structure influences function.
Wait, so
Yeah.
cellulose is like straight, right? So why isn't that easily digestible when enzymes can reach it fast since it's straight?
Good question. Oh, no, that's not what I want to write. Structure influences function. Thank you for that. She started talking to me.
Oh,
Not your fault. I'm writing, talking, listening, thinking, and it just messes me up. Plus, I'm a bad speller. Add all that together, I'm going to make a lot of mistakes in the videos. And then I have to deal with him. So yeah, cellulose is what kind of linkages in cellulose? So it's beta what? 1,4. They are straight. They form these long, thin fibers. Cellulose is, when you say a high fiber diet, what they're talking about is fiber. That's fiber. Because it literally is, when you think of fiber, when you look at the strings that make this up, it's made of long, straight chains of cellulose. Yeah.
What's the difference between alpha and beta linkages?
So you want me to go into that? I was hoping not to get into that. But okay, no, it's going to be, if it's a question, it's a question. I want you to understand. So I'll do it. I'll show you. But for now, can you get the idea that this is a straight chain? And her question is, look, this one's harder to digest because it's all curly and it's hard to get at it. But there's a second part you're not thinking about. You're right that these would be easier to get at because they're straight. And they're not even branched, they're straight chains. The problem is we don't have an enzyme to break them up. We can't digest it. It can be as straight as you want. You can lay out one single fiber and put it in my belly, inside my stomach, and it'll sit there because we have no way of breaking it down. We have no enzyme that'll help us break it up. It's very tough to digest this. The only organisms that have this ability are fungi and bacteria. That's it, decomposers. You might think, what about cows? They eat grass and they have bacteria in their bellies. What about termites and what else eats wood? Well, termites are the only ones that come to my mind. But what about termites? They have bacteria in their digestive system. Is that making sense to y'all?
Yeah.
So theoretically, if you had those bacteria in your belly, then you could, but you wouldn't want to do that. Yeah?
Is blood sugar like literally the amount of sugar that's in your blood?
Yes. Yes. It's a concentration of sugar in your blood. Good question. Absolutely. Glycogen. What kind of linkages are in glycogen? Alpha what? 1, 4, and 1, 6?
Yeah, it's alpha 1, 4, 6. And it's branch.
So let's take a look, and it's of course that again is branched. And this is straight. Now, that's what makes it highly digestible. It also needs a lot of space, and it needs a lot of water. It holds a lot of water. So your liver has a lot of this sugar extra sugar you eat. If you abuse that sugar, not only do you have the problem with diabetics, but you can get fatty liver disease. You have giant fat deposits that start developing inside the liver. You'll actually see it in an X-ray or CAT scan. And if you let it go too long, you'll start to get cirrhosis. So high sugar can actually give you cirrhosis of the liver. Holes in your liver that impact your function. You'll start to see liver function go down, and then it's a liver transplant or death. It's not like you, at least with kidneys, you can go a few months. Without your liver, you can't, because you can do dialysis. Without your liver, you start to turn yellow, and it's called jaundice, and then it's over. So
babies have jaundice when they are
that's different. That's a that you understand. So now that we've had time to look at it, the videos on the big screen, and I think people took pictures of the screens, of the screen picture. If you want that, could you send me that link as well? I'll send it off. Somebody else showed me Fiveable. It's a really great Fiveable, it's called. It's, I think, is there an E in Fiveable?
Yeah.
Well, I don't know if they do. It's a website. It's a trademark. I don't know what they do, how they spell it. So Fiveable is a website that it's actually a book series as well that goes through and summarizes. Remember I told you the Cliff Notes? It's like the Cliff Notes. But as I warn you, Fiveable it does a good job with vocabulary and it lets you play around with summaries. It's useful, but if you haven't read or done kind of work through the details, it's not as useful as it could be if you read first and then went back. But it's very good for studying. It pretty much makes your study cards for you. I'm going to warn you, the best way to make study cards is you make them. Write them down and study them. But this is it's a good website. It does show you what's important. I know some people are having trouble in that activity, what's important or what's not. Well, this is what's important about or not about everything.
Does this cost money?
No, Fiveable online is pretty free. I
It's
mean,
free unless like there's certain stuff that costs money.
but this general stuff is up there. There's a lot of this out there. You will find apps for your phone too that will give you summaries.
Wait, I'm trying to find something. Where's, like,
There are apps online. I'll send them. I'll send you all the links I have. I have a ton of links if you want. So these are your linkages. These are your linkages for each of these. These are what we call the polymer stages of sugar. So this is sugar as a polymer.
Mendoza.
Yeah.
You're writing and nobody can see it because this is a desert and it's not public. So.
It's a desert.
Look.
It doesn't care.
Oh, Lord.
Did you get what I'm saying?
What was that? Explain to us.
No, I can't because
Why not? He's trying to hold back.
I've only gotten through 20 minutes of class. So, Fiveable is a good resource, and yes, I would love to do a Kahoot before the quiz. Fine. And we could do a Kahoot tomorrow before the quiz. There you go. That's it. Okay? So, but guys, specifically, then you're looking at polymers and monomers. When we're talking about polymers of sugar, you're talking about 1, 2, 3, and 4 got jumped up. I don't know why. 4 jumped up here. So, and 4. Those are the 4 big polymers you need to know, and you need to know their linkages so that you can... What is going on? 4. As Jonathan loves to say, you can't say that. So 1, 2, 3, 4. We have these 4 main structures that we call polymers of sugars. You need to know what they are and what they're used for. And the reasons why they're used that way, right? So glycogen is branched. That's really helpful to fill in, get as much space in the liver to store energy, to store sugar as you can. Cellulose is straight chain. That allows it to be stacked and held together really well. And especially if you crisscross them, it's really good at holding structure together. That's what allows trees to grow so tall and be so strong, why we use wood for building houses, because the fibers are all lined up. These are fibrous. Starch is used for storage of energy in potatoes and yams. And amylose is curled up. It's straight, but curled is straight in the sense that it's a long chain. But the chain is curled up, where amylopectin is curled up and it's branched. And so glycogen is branched, and it's good that it's branched. Now, that being said, when we look at the dimers of sugar, the disaccharides are going to be important as well. And some of the important ones that we talked about are lactose, important for mammals anyways, because mammals are the only ones that make it. They're the only ones that lactate because they're the only ones with the glands to do it. They're not the only animals to make milk. There are ants that milk slugs for food. They actually farm little insects for food, the larvae of other insects. They farm fungus and eat the fungus as well. Ants are very interesting. That's why I hate to hear people killing them for no reason. So another disaccharide is sucrose. Another disaccharide is what? What's another disaccharide we talked about? Galactose. Is galactose a disaccharide? I'm asking a question. No, it's not. So what's a disaccharide? Fructose is a monomer as well. So another one we talked about is maltose. I know. I was like, how many is she going to go through? Oses is she going? I keep saying that ose at the end is sugar, so it's good that you're getting a lot of oses. So that's good. Sucrose is table sugar. Maltose is found in malt drinks and You've heard of malt milk,
Malt,
malted milkshakes, Yeah. Those little balls, those little whoppers also have malt. It's all made of this kind of sugar. Lactose is obviously in milk. So some examples. You should know some monomers at this point. And some of the monomers are obviously glucose is a monomer. And someone said fructose is correct. So you have fructose, which is a 5-ring structure, not 6-ring, but there is 6 sugars. Right? There are 6 sugars. It's just that it's only 5 members in the ring, and you built it so you know it. You know both of these, where this is fructose. Galactose, yeah, I'm not going to test you on that one too much because I don't really think when it comes to the big picture of trying to get to the AP exam, focusing on that. Galactose is important. It's in lactose, so yes. And yes, it's a 6-carbon sugar, a 6-sided sugar as well. And it is another structural isomer of glucose. It's called galactose. And we use the idea of maple syrup urine disease and the issue with being able to digest galactose. But when we look at fructose, is it?
Sorry for interrupting, but we went through a program, Horizons, yesterday, and the med student told us that they didn't actually taste the urine for maple syrup urine disease. They smelled it, and it smelled like maple syrup.
Okay, the med student's wrong. I'll pull up the ancient Greeks.
You were wrong.
I know, we're all there. But I will, the med students, could I be wrong? Yes,
No. She said
it's possible.
She said they test urine for diabetes, right? Yeah, they taste urine for diabetes.
blood test. I mean, urine test.
Urine test.
And the
They taste
HbA1c
it.
test.
But in the olden days, they taste urine for diabetes. It's what we're told.
Yes, they tasted urine for diabetes. If it tasted sweet, they knew people had diabetes. They called it wasting disease. The ancient Greeks, the first, I forget his name. I think the father of medicine might have been the guy that did it. The guy with the staff, with the 2 snakes. The ancient Greeks, I'll do the research. I'll print it and bring it to you. I'll bring it to you. I want to have the references for you. I'm going to move on. I'm not going to. It could be wrong, but I don't think so. What? What? What did she say? Oh my God. See, that's what I'm trying to tell you. We got to stick to one thing at a time. You're jumping around, and then you're saying you're confused because you're jumping around. Stick to one thing at a time. So, hexose, these are hexoses. Why do we call them hexoses? Because they have 6 carbons. They have 6 carbons. They have 6 carbons. That's why we call them hexoses. There are also these other types of sugars that are pentoses, which are going to have how many carbons? 5 carbons. The only one really, the only 2 that you're going to have to worry about is this sugar called, that's 1, 2, 3, 4, 5. Can you notice the structural similarities? You have a ring, and there's one sticking out. It's a lot like fructose, except fructose has this one that sticks down. Just this simple little chain difference of having one missing carbon. In any case, this pentose is called ribose. It's called ribose if, like all other sugars, for every carbon, you have an OH group on it. Now this one I believe is missing the OH. 1, 2. If there's an OH here, and this would obviously have an OH sticking out over here. If there's an OH here at this is the key difference between ribose and then what's the other one I'm gonna draw? Can anybody guess?
Deoxyribose.
That's right, deoxyribose. The main difference is that in deoxyribose, the OH is gone and in its place is a hydrogen. And that difference determines a lot. That difference determines a lot. Ribose versus deoxy. Deoxyribose. All right? And they're both pentoses. One's going to make DNA. It's going to be the backbone of DNA. Sugar phosphate backbone. This will not be the last time I say it. Where this is going to make RNA, ribonucleic acid. And the reason they call it nucleic is, I hope you know why, but if not, we're gonna make nucleotides, but the key is nucleic because an acid, it's nucleic because it has to do with the nucleus, right? Remember, ic, as one of our colleagues in the room made sure we understood that anytime you have itic, it means it's involved with. And so when this is standing up here, you have a nitrogenous base, you have a phosphate, you'll have a nucleic acid. All right. So that's the stuff that's going to be on the test. And one last thing, glycosidic bonds. Let's remember when you have two sugars, I'm going to go ahead and do the one we just saw on the board. When you have a glucose, one OH, both the OHs are going to be down. And what is it? What's going to be the... let me just do it, no see it all. This being a really bad drawing of glucose, there's two of them. What is it going to be? 1, 1? 1, 1? 1? What are the linkages? 1? Well, these are both down. 1, 4. So if this is down, and this is 1, 4, so 1, 2, 3, 4. This is going to have an OH as well. Well, it could be anything, any glucose. This is going to be any OH down 1, 4 linkage. What's OH down? Alpha or beta? Alpha. So any alpha 1, 4 is going to do this, and it's a glycosidic bond, so that means that what you're going to get is one of the H's and one OH is going to come out. And what are you going to do with that extra bond?
Dehydration.
Well, this is dehydration synthesis, but what is going to happen is you're going to do what?
H2O.
Water's going to come out. That's part of it.
It would be bound by O.
And then the synthesis part.
Is O.
So the dehydration is H2O's coming out, correct. The synthesis part is that oxygen, the extra bond, is now going to connect and make something new. I had a monomer. I had 2 glucose monomers, alpha glucoses. When I'm done with an enzyme that helps me do this, enzymes are required to do this, in the end, I have 2 of them linked by a glycosidic bond, and that's your glycosidic bond. Anytime you have an oxygen link between two sugars is called the glycosidic bond because there's sugar, water's coming out, so it's called dehydration synthesis. All of our macromolecules are going to be the same. And why don't I just pull up a Kahoot about macromolecules? Because we haven't talked about proteins yet, nucleic acids yet. As one of your colleagues looked up, what about nucleic acids? Well, we haven't gotten there yet, right? We're still working on alpha versus beta. So that's what you need to know for the quiz tomorrow. If you can find a Kahoot, if you all find a Kahoot that can go over just that before the quiz, I will let you play it in class before the quiz for practice. Or you can make one, you can make a Kahoot, or you can find one. All right? And you can play it. I'll sit there and watch it. I'll even correct anything you make. But I'm not making one tonight. I'm not doing that for you. Yeah. You can. Anybody make it? And can you make extra credit? You want to make an assignment, extra credit assignment? We can. And you guys can even combine them later in the morning up. Or play multiple ones. But we have to get the quiz done tomorrow for sure. Are we all good with this?
Yes.
All right.